Welcome, everyone. Today, we're unpacking a piece of technology that is fundamentally changing how we understand the genetic building blocks of life on Earth. We're going to dive right into how genotyping by sequencing, or GBS for short, is completely revolutionizing DNA analysis and plant breeding to help solve massive global issues like food security. I want you to think about this number for a second: $5. Back in the early 2000s, the cost of sequencing a single human genome was in the millions. Millions. But to feed a rapidly growing planet, we simply can't be spending millions, or even thousands, every single time we need to sequence crop varieties. This $5, this represents the incredible per-sample cost drop we've achieved today, all thanks to the GBS method. And this brings us to a mind-blowing breakthrough in molecular biology. Seriously, how do we read the DNA of up to 384 distinct plants simultaneously, all for the price of a fancy cup of coffee per sample? Well, it happens by combining the raw power of next-generation sequencing with some incredibly clever biological multiplexing. So here's the crucial takeaway. GBS isn't just cheap. It's a completely streamlined way to discover single nucleotide polymorphisms, or SNPs, across an entire population. And honestly, the truly revolutionary part here is that you can often pull this off without even needing a prior reference genome for the species you're studying. It works by essentially reducing the massive, overwhelming complexity of a genome using specific restriction enzymes, which lets us generate high-resolution data really fast and incredibly efficiently. Here is our quick roadmap for today's explainer. 1. The DNA sequencing bottleneck. 2. How GBS actually works. 3. Real-world impact on plant breeding. 4. The bioinformatics data challenge. And 5. The future of genetics. All right, let's jump right into Section 1, the DNA sequencing bottleneck. To really appreciate this, we have to look at how things used to be. In the past, sequencing relied on mapping individual genomes at massive expense. Scientists were out there painstakingly resequencing tiny panels of parental individuals just to find a few variants. It was incredibly slow. Today, the GBS paradigm completely flips the script. It enables us to simultaneously sequence hundreds of individuals within a population all at once. We're now using those sequence differences directly as genotypic markers, which totally changes the scale at which scientists can operate. Moving on to Section 2, how GBS actually works. So, what's actually happening in the lab? The GBS protocol kicks off by plating out the DNA. Then, it uses methylation-sensitive restriction enzymes, a super popular one being APK. Now, this is a brilliant biological trick. By using this specific enzyme, researchers can basically skip over all the highly repetitive, uninformative junk regions of a genome. Instead, it acts like a heat-seeking missile, targeting the important lower copy regions with 2 to 3 times higher efficiency. After cutting the DNA, barcodes and adapters are ligated, or attached, to the ends of these fragments, and then PCR is used to amplify the whole thing. Now, here's where the multiplexing magic happens. By assigning short, totally unique DNA sequences, which we call barcodes, to each sample in a 96-well plate, every single sample gets its own biological ID tag. Because they all have name tags, essentially, you can take all 96 samples, or even up to 384 lines, and pull them into one single sequencing tube. The sequencer reads the whole batch at once, and a computer later sorts everything out by their barcode. This one step is exactly what drastically slashes the cost down to $5. The advantages here are an absolute game changer. You've got fewer purification steps, and you completely eliminate the tedious process of size fractionation, so the workflow is incredibly fast. But the biggest win? Because you don't require a pre-existing reference genome, researchers can just jump straight into analyzing highly diverse or even completely novel, undocumented species. Let's look at what this means on the ground. In Section 3, real-world impact on plant breeding. Did you know that over a third of the world's rice is actually grown in really poor, low-fertility soils? It's a huge problem. So researchers use GBS to evaluate wild introgression lines of rice, specifically derived from a rugged wild species called Oryza rufigon. They wanted to test these lines across different environments to see exactly how they survive severe nutrient stress, specifically low phosphorus conditions. By running this wild rice through the GBS pipeline, scientists didn't have to just guess which plants were doing better. They precisely mapped out 113 distinct quantitative trait loci, or QTLs. In plain English, these are specific genetic regions linked directly to crucial survival traits, things like days to flowering, overall plant height, and most importantly, grain yield when the plant is under extreme stress. The huge payoff here was that GBS allowed them to pinpoint a specific hotspot right on chromosome 1. Right there, they found the O's SPT11 gene. This is a dormant trait from wild rice involved in phosphorus acquisition. Basically, it acts as a switch to activate a beneficial symbiosis between the plant's roots and the surrounding fungi in the soil microbiome. And thanks to GBS, this ancient hidden survival trait can now be bred into modern rice crops to make them incredibly resilient to poor soils and our changing climate. But it's not all perfectly smooth sailing, which brings us to Section 4, the Bioinformatics Data Challenge. So as the physical sequencing got cheaper and cheaper, the real cost burden just shifted down the line. We moved away from the lab work, which, as we know, is now cheap, efficient, and highly multiplexed, straight into a massive computational bottleneck. These sequencers are spitting out unimaginable amounts of messy, complex digital data that require intense data management and heavy-duty bioinformatics pipelines just to interpret and store. You see, because GBS relies on low-coverage sequencing to save all that money, meaning it's essentially taking samples of the genome rather than reading every single letter perfectly, it inevitably produces missing reads. This leads to missing genotypes, sequencing errors, and something called undercalled heterozygotes. That's just a fancy way of saying the computer might mistakenly think a plant has 2 identical copies of a gene when it actually has 2 different ones. To combat this mess, scientists use various software pipelines. But get this, the proportion of missing data can vary wildly depending on the software you decide to use. It can range from 28% missing data with Tassel GBS all the way up to over 57% missing data if you process the exact same sequence through the Stacks pipeline. Choosing the right software has literally become just as important as the biological chemistry itself. So how do we fix the gaps? Well, researchers certainly don't just throw the data away. They implement highly advanced data imputation methods to essentially fill in the blanks mathematically. They filter out errors by looking at minor allele frequencies, they double-check against known parental genotypes, and yes, they still rely on careful manual curation. All of this ensures the final genetic selection models are highly accurate and ready for real-world breeding.
Which leads us to our final section, Section 5, the future of genetics.
I absolutely love this quote from the source material. Soon, plant breeders may conduct genomic selection on a novel germplasm or species without first having to develop any prior molecular tools. I mean, that perfectly captures the sheer scale of the potential here. We have the ability right now to analyze and protect the biodiversity of practically any species on Earth without needing to spend decades of preliminary research just to build a reference genome first.
The genetic floodgates are officially wide open. The applications here stretch from discovering drought-resistant wheat and low-phosphorus rice to establishing completely new, revolutionary understandings of animal conservation. We are stepping into an era where understanding the very code of life is faster, cheaper, and more accessible than it has ever been in human history.
Thank you so much for joining me on this explainer today. As we've seen, this technology represents a colossal leap forward, which leaves us with one final, thrilling question to ponder. If we can unlock the DNA of practically any species for just $5, what dormant, climate-resilient traits will we discover next? Please don't forget to like, subscribe, and visit our website to keep exploring the fascinating world of genomics with us. Until next time, stay curious.